Hello everyone, welcome to Clarity for Sure. Today in this video we are going to discuss the MCQs on the important topics related to biotechnology, which is helpful for your upcoming UPSC CSE Prelims examination. This MCQ series is based on the lecture that I had taken yesterday on the same topic, that is the important topics related to biotechnology. So without any further delay, let's get started. Now, the question number 1. With reference to nucleic acid, consider the following statements. The presence of hydroxyl group on the second carbon of the sugar in RNA contributes to its catalytic activity and lower stability. In eukaryotic cells, both DNA and all types of RNA are synthesized inside the nucleus. Uracil in RNA replaces guanine to allow efficient base pairing with the adenine during transcription. Okay, these are the three statements, and the question is which of the statements given above is or are correct. Option A 1 only, option B 1 and 2 only, option C 2 and 3 only, option D 1 and 3 only. So the answer for this particular question is option B 1 and 2 only. See, when we were discussing about the difference between DNA and RNA, I told you that the presence of hydroxyl group in the RNA actually lowers the stability. So statement 1 is correct. And second point is, we all know that in the eukaryotic cells, both DNA and all types of RNA are synthesized inside the nucleus. So do not get diverted because of the statement where it has given all, because sometimes the extreme statements become correct. Okay. Third, uracil in RNA replaces guanine. No, the uracil will replace thymine in, not guanine, in RNA to pair with adenine. Okay. So the answer for this particular question is option B 1 and 2 only, as statement 3 is wrong. I hope this MCQ on the concept of the nucleic acid which we are discussing here is clear to you now. Moving to question number 2. Which of the following best explains why DNA is more chemically stable than RNA? Your options are DNA as uracil, which enhances hydrogen bonding. DNA lacks phosphate backbone, reducing reactivity. DNA contains a deoxyribose sugar lacking a second hydroxyl group. DNA is confined to the nucleus and hence avoid enzymatic degradation. So the answer for this particular question is option C, because we have seen in the yesterday's video where I have clearly mentioned when I was discussing the difference between DNA and RNA, where because of the lack of this OH group only, DNA is less prone to hydrolysis. Okay. Hence, it increases the chemical stability, whereas in RNA, the hydrolysis takes place. That is why it is less stable. Okay. So the answer for this question is option C. Moving to question number 3. Which of the following correctly explains why mRNA must be relatively unstable in the cells? It is talking about the messenger RNA after the transcription stage. Okay. Stability ensures prolonged synthesis of a single protein, preventing overproduction. Instability allows for rapid turnover and tight regulation of gene expression. Instability enhances accuracy of base pairing during transcription. mRNA degradation is essential for transcription to occur. See, here it is an extreme statement. So you can eliminate option D. Okay. Now we should be able to choose between the rest of the options. The answer for this question is option B. Why? Because the mRNA must be relatively unstable. Why? Because this mRNA instability will actually allow the cells to rapidly adjust the protein production in response to the environmental signal, which is a key feature of a gene regulation. Okay, this instability of mRNA actually adjusts the protein production in the next stage, that is the translation stage, which is a very important part for gene regulation. So once this protein is no longer needed, okay, after a certain point of time when the protein production is not needed, this mRNA is quickly degraded to stop the further protein production. Why this protein production, the stoppage of protein production becomes important? Because this protein production enables the cell to respond efficiently to the environmental changes, stress and development signals. Okay, so at till a certain point of time the protein production is needed. Once the protein production is not needed, the mRNA will stop. It will get degraded, because we know that with the mRNA, because mRNA is the protein where it contains the genetic information from the DNA. So if the mRNA is degraded, the further protein production will stop. Why the protein production will stop, and what is the use of this protein product, stoppage of this protein production? This enhances the gene regulation and to adapt itself for the efficiently to the outside factors like environmental changes, stress or development signals. So that is why this mRNA instability, the relative instability is very important. Okay, I hope the concept behind mRNA instability is clear to you, and why mRNA must be relatively unstable is also clear to you. Moving to question number 4. With reference to the genetic modification techniques, consider the following statements. Recombinant DNA technology can be used to insert a gene from one species to another, even across kingdoms. Gene editing to CRISPR can only add new genes but cannot remove or correct existing genes. Protoplast fusion requires removal of the cell wall to allow two plant cells to merge. So, which of the following statements is or are correct? Option A 1 and 3 only, option B 2 and 3 only, option C 1 and 2 only, and option D 1, 2 and 3. That is the all of the above. But the answer for this particular question is option A 1 and 3 only, because we easily know we know that this CRISPR-Cas9 or CRISPR-Cas12, whatever technology that is behind this CRISPR, can do both removing, editing or to correct the DNA sequence. It is not just to insert, but it can also make some changes, remove, edit or correct. So statement 3 becomes wrong. So if you eliminate statement 3, you are left, statement 2 is wrong. So if you are eliminating statement number 2, you will be easily able to arrive at the right answer. That is option A 1 and 3 only. I hope this genetic modification technique and the question related to this is clear to you now, and I hope the option elimination will also be clear to you. Coming to the next question, consider the following statement regarding gene knockout and gene silencing. We have gone through these terms in the yesterday's session. Gene knockout involves permanent alteration of a genome, while gene silencing is generally reversible. Both techniques can be used to study gene function, but only gene silencing uses RNA interference mechanism. Gene knockout and gene silencing always result in the complete loss of gene function. So pay attention to the extreme words always, only. Okay, so please pay attention to these words. Answer for this question is option number A 1 and 2. Why? Because gene knockout and gene silencing may not lead to complete loss of function. It actually reduces or it will block the expression either temporarily. Okay, but in gene knockout we can permanently edit that, but not in the case of gene silencing, whereas gene silencing is a temporary changes that we are making to that particular DNA sequence. Okay, it is to study that particular DNA molecule by silencing a part of gene, how it will behave without that particular gene. So gene knockout is a permanent one, but gene silencing is not a permanent one. So if you are paying close attention to this particular statement always, you will be able to eliminate option number 3. So you will be easily able to arrive at the answer, that is option A 1 and 2. See here, you can see this only statement. See, if you have paid attention to the yesterday's session where I was discussing about the gene knockout and gene silencing, I clearly told you that there is an RNA interference mechanism in the gene silencing, because that RNA will act as a guide RNA. What is guide RNA? It will take that particular nucleoside to that particular place, the exact place of that DNA where a particular gene or where a particular enzyme has to be deactivated. So for that, this RNA interference, which will act as a template, becomes important. Okay, so I hope the concept of gene knockout and gene silencing is clear to you now by this particular question. Okay, coming to the next question, with reference to the advanced plant biotechnology, consider the following statement. Protoplast fusion can lead to hybrids that would be difficult to obtain through traditional breeding. Recombinant DNA and CRISPR techniques cannot be used in plants due to the cell wall barriers. Gene silencing is often used in agriculture to develop disease resistant plant crops. See, if you pay close attention to this particular statement, number 2 cannot be. We know that recently this CRISPR Cas9 technology was in news because India was the first country to release two genetically edited rice. Okay, so if you are aware of that particular news, you could easily eliminate statement number 2. So if you are going to eliminate statement number 2, the answer will be option B, that is 1 and 3 only. Yes, the answer is option B, because we know that both recombinant DNA and CRISPR are widely used in plant technology, biotechnology, despite cell wall, often after techniques like agrobacterium mediated transfer or biolistics. Okay, because we know that this CRISPR-Cas9 technology, I hope you would have watched the video where I have dealt with this CRISPR-Cas9 and CRISPR-Cas12 technology, where a guide RNA will be guided that particular nucleoside to that particular place where an editing or slicing of a particular enzyme is done. Even despite the cell wall, this CRISPR-Cas9 is efficiently able to do the gene editing in the plants also. So that is why the statement number 2 is wrong. If you are aware of the news which I was telling related to the genetically gene edited rice, you would be easily able to eliminate option number 2. So the answer for this question is option B, 1 and 3 only. I hope the CRISPR-Cas9 and the recombinant DNA is clear to you now. Coming to question number 7. Which of the following statement best distinguish gene editing from recombinant DNA technology? Gene editing is limited to plants while recombinant DNA works only in animal. Extreme statements. Gene editing alters existing sequence while recombinant DNA introduce foreign genes. Both techniques work only by inserting plasmids into host cells. Gene editing is reversible while recombinant DNA is not. See, we can easily eliminate option C and option A because there is a use of extreme statements, because in the previous question only we saw that both this recombinant DNA and gene editing applies to both plants and animals. Okay, both techniques work only by inserting plasmid to the host cell. No, the statement is wrong. What is the answer for the question then? It is option B, because we know that gene editing alters existing sequence while recombinant DNA introduce foreign genes. Yes, CRISPR-Cas9, for example, in the gene editing can precisely modify the genes, while recombinant DNA will insert a new genetic material, that is across species, across kingdoms, but that is the basic difference between the CRISPR technology and the recombinant DNA. Okay, and regarding question number option number C, we know that recombinant DNA often uses plasmids as vector, but CRISPR uses guide RNA and Cas9 protein, which I told you already. This guide RNA will act as a template and it will guide this Cas9 protein to the specific site where a gene editing has to be done. This Cas9 protein will act as a molecular scissor and will cut or remove or edit that particular gene. Okay, and that is why it does not rely solely on plasmids, and we all know that gene editing is reversible while recombinant DNA is not reversible. Okay, so the see, this statement is also wrong because both are irreversible. Okay, so the answer for this particular question is option B, alters the existing sequence while recombinant DNA introduce foreign genes. So both are option A, C, D and are wrong, and option B is the correct answer. Question number 8. With reference to the genetic modification in plants, consider the following statements. Recombinant DNA technology introduced specific genes from unrelated species using vectors, which we saw just in the previous slide, uses plasmids, specifically the vectors. Protoplast fusion always results in genetically modified organism due to genome mixing. If you had remember, while I was discussing the difference between the recombinant DNA and protoplast fusion does not always result in genetically modified organism, because it is often debated whether protoplast fusion is GMO or not. Okay, so you can eliminate option number 2 easily. If you are going to eliminate option number 2, you will be arriving at the right answer. Option B is 1 and 3 only. Recombinant DNA and protoplast fusion differ in that the former alters the specific gene while the later merges entire genomes. I hope you would be remembering the difference again when I was talking about the alteration that is being done in recombinant DNA and protoplast fusion, where recombinant DNA, where we insert a specific gene from a different species across kingdoms, whereas in protoplast fusion we will be taking a very two good traits of two plants. Say one is a drought resistant plant and another is an high yielding variety, but this high yielding variety is not drought resistant, this drought resistant is not high yielding variety. So we are going to take two genomes of these two types and we are going to make a new genome. Okay, the entire genome of this and this. So that is the basic difference between the recombinant DNA and protoplast fusion. So the answer is option B. Protoplast fusion does not always produce GMO. It fuses cells closely related species, which I have told you already. And since no foreign gene is inserted, whether it's a GMO is still debated. We have discussed the same point in the lecture also. I hope the difference between the recombinant DNA technology and the protoplast fusion is clear to you now, where in the recombinant DNA we will be focusing on a specific gene, whereas in the protoplast fusion we will be merging the entire gene of this high yielding variety plant and the drought resistant plant, so that we will be getting a new type of getting a new plant which will be having both the traits combined. Okay, I hope the difference between the recombinant DNA and the protoplast fusion is also clear to you. Coming to question number 9, consider the following statements regarding the recombinant DNA and recombinant protein. See, recombinant protein is the product of recombinant DNA. Okay, recombinant DNA is a genetic material produced by combining genes from different organisms, while recombinant protein is a protein produced from expressing recombinant DNA in the host organism. Recombinant DNA is used to produce drugs, enzymes, and vaccine, while recombinant protein is used only in the genetic research. So please pay attention to the extreme words that are being given in the statements. The process of creating recombinant DNA involves transcription and translation, while recombinant protein production involves gene cloning and restriction enzymes. So we will for now try to eliminate option number 2. So we are left with only one option, that is option number A. So see, even if you are not aware, even if you are not sure whether to eliminate the second or third statement, first try to eliminate the statements which you feel are surely wrong. Okay, so I am eliminating option number 2, but so luckily I am arriving at a single option, that is option number A. Now we will see whether this answer is correct or not. Yes, the answer is correct, because recombinant protein is used in medicine and industries like insulin, vaccine, and not just research. Why statement number 3 is wrong? Because recombinant DNA is where it uses the gene cloning and restriction enzymes, whereas the recombinant protein production involves the transcription and translation after gene insertion. I hope you are aware of, you are still remembering the gene regulation which we were discussing yesterday, where I was talking about two stages, transcription and translation. In the transcription stage, the DNA will be converted to mRNA, only in the translation stage the mRNA will get converted into protein product. How this mRNA will conduct the genetic instruction that is being given by the DNA molecule. So based on that only the protein synthesis the protein synthesis will occur after the transcription stage, that is in the translation stage. Okay, so if you are clear with your basics, you will be able to eliminate option number 3 also. See, even though if you are not aware of a particular thing, but if you are aware of the basic terminologies related to the DNA, RNA, transcription and translation, you will be able to eliminate the options, easily eliminate the statements from the questions. Okay, I hope question number 9 is also clear to you now. Question number 10. Consider the following statements regarding gene regulation and mRNA. Transcriptional regulation controls the production of mRNA from DNA, acting as a main on or off switch for gene expression. Post-transcriptional regulation involves modification like splicing or RNA stability before the mRNA is translated into the protein. Translation regulation focuses on modifying protein after they are synthesized, like activation and deactivation. If you were clear when I was talking about this activation or deactivation, I was discussing about 4 stages in gene regulation. Okay, in the 4th stage I was telling that only in the post transcriptional stage the post transcriptional regulation only focuses on modifying proteins after they are synthesized, like activation or deactivation, but not the translation stage. So if you are aware of that particular points, the levels which I was discussing yesterday, you will be able to eliminate option number 3. So if we are eliminating option number 3, the answer is option A, because I told you that post translation regulation is responsible for modifying the protein after it is synthesized, because we know only in the translation stage the protein will be synthesized. So how the protein has to be activated or deactivated should come after this translation, that is the post translation. So I hope this difference is also clear to you now. So the answer for this particular question is option number A. Okay, 1 and 2 only, as statement 3 is wrong. I hope this mRNA and the gene regulation, the stages, 4 stages which we were discussing, transcriptional, post-transcriptional, translational and post-translation, are clear to you now. I request you all to kindly watch the video where I had discussed about all these basics in the separate video, so it will be very useful for your exam coming Sunday, so that if you are getting any question related to this biotechnology, you will be able to answer these questions with ease. Okay. Coming to question number 11, consider the following statement about transcription and translation. See, transcription and translation, I hope you are aware of the differences between the transcription and translation now. Transcription occurs in the nucleus in eukaryotes and in the cytoplasm in the prokaryotes. Translation takes place in cytoplasm, where ribosomes decode mRNA to build protein. During transcription, RNA polymerase synthesizes mRNA based on the DNA template, while in translation, ribosomes decode mRNA using tRNA. Okay, so which of the following statements are correct? Option A, 1 and 2 only. Option B, 1, 2, 3. Option C, 2 and 3 only, and option D, 1 and 3 only. The answer for this question is all the statements are correct, because it is a very direct question. We all know that the transcription occurs in the nucleus of the eukaryotes, whereas it occurs in the cytoplasm of the prokaryotes. Okay, translation takes place in cytoplasm, where we know that ribosome is the enzyme that will decode the mRNA to build proteins in the translation, whereas in transcription the RNA polymerase synthesizes the mRNA based on the DNA template, while in translation ribosomes decode mRNA using tRNA, that is the transmitter RNA. I have discussed this in detail in the video that I have posted yesterday. That is the difference between the transcription and translation. Transcription is nothing but the conversion of DNA to mRNA, whereas the translation stage, this mRNA will be converted into the protein products. These protein products are converted based on the genetic information of DNA that is that is carried by this messenger RNA, that is the mRNA, which is a temporary RNA. Okay, so the answer for this question is option B, 1, 2, 3. Okay, see, sometimes when you are going to read a question, this would sometimes look like unfamiliar, but if you are applying your basics to this particular question, it will become very easy to you. Okay, so when you are in the exam pressure, the mind will not work properly, but if you are paying close attention to a particular question, you will be, the chances of getting that questions right will always be high. Okay, having discussed the question number 11, we'll go to question number 12. What is the role of base pairing during transcription and translation? It is talking about the nitrogenous bases both in the transcription and translation. This is a very direct question. The answer for this question is answer option A, because in transcription the base pairing will be AT and GC, because I told you this earlier also, because these nitrogenous bases will be occurring in pairs only. So it is adenine and thymine, it is guanine and cytosine in the translation, whereas the mRNA codons match with the tRNA anticodons, which this also I have discussed in the slide where I discussed about the transcription and translation. So in the base pairing, that is in the DNA, the transcription of from DNA to mRNA, the base pairing will be AT and GC, whereas in translation the pair will be mRNA codon and tRNA anticodons. This is a very basic thing. Okay, now coming to question number 13. With reference to the recently developed next CAR-19 CAR T cell therapy, consider the following statements. CAR T cells are generally engineered white blood cells. They are designed to target and destroy cancer cells. This is India's first indigenously developed CAR T cell therapy, developed with support from IIT Bombay. The therapy involves modifying patients' red blood cells to express synthetic receptors that can recognize the tumor antigens. CAR T cell therapy is particularly effective in treating certain types of blood cancers like leukemia and lymphoma. Okay, see, if you pay close attention to statement number 1 and statement number 3, statement number 1 talks about the white blood cells, whereas the statement number 3 talks about the red blood cells. We all know that the T cell is the WBC, which is cytotoxic in nature. So if we are going to eliminate option number 3, we are left with two options, 1, 2, 4, 2 and 4. And we all know that, see, these are genetically injured white blood cells that are designed to target the destroy cancer cells. So statement number 1 is correct. So the answer is option number A, 1, 2 and 4. Okay. So coming to the next question. With reference to recent advance in biotechnology, consider the following statements. CRISPR-Cas9 is a gene editing technology that can be used to modify the DNA of living organism with high precision. Genetically modified organisms developed using gene editing technique like CRISPR-Cas9 are currently exempted from regulation under India's Environmental Protection Act 1986. India has approved the commercial cultivation of genetically edited variety of mustard developed through CRISPR technology. So, if you pay close attention to this particular third question, genetically edited, is the mustard genetically edited? No, it is genetically modified. It is not genetically edited. So, eliminating option number 3, you are left with option A and option B. So, the answer for this question is option A. Why? Because in India, GMOs, including those developed using CRISPR-Cas9, are regulated under the Environmental Protection Act. Only the gene-edited crops, and I have clearly explained the difference between the gene editing and genetically modified organism in the lecture where I was dealing with the gene editing of the two crops that were released recently, two rice varieties that was released recently. So kindly watch that video, so you will get the basic understanding of the difference between genetically edited crops, gene edited crops and genetically modified organisms. Okay, so statement number 2 is also wrong, statement number 3 also not, because this mustard is not, the Dara mustard is not genetically edited, it is genetically modified. Okay, so the answer for this question is option number A, that is 1 only. I hope I hope the concept behind this is also clear to you now. Kindly watch the video where I have discussed the difference between GMO and genetically edited gene editing. Okay. Question number 15. With reference to triocell DNA, consider the following statements. TDP1, consider the following statements. TDP1 is a DNA repair enzyme that helps fix damage caused by chemotherapy drugs as camptothecin. Inhibiting TDPN in cancer cells may enhance the effectiveness of chemotherapy by preventing DNA repair. Overactivity of TDP1 in cancer cells can make them more sensitive to chemotherapy. It is not more sensitive, it is more resisting, because I hope you remember the concept of DNA repair and the role of this TDP1 enzyme, where this is the main culprit, where it actually repairs the DNA of the cancer cell that is being attacked by the chemotherapy. So due to this self repair thing of using this TDP1 of that particular DNA, in that particular DNA the treatment the cancer cells become resistible to the treatment. So scientists have decided to target this TDP1 enzyme, so that once this enzyme is edited using the CRISPR-Cas9 and whatever technology, or silencing, gene knockout or gene silencing, whatever we can do this particular thing to this TDP1, this will become the treatment for the cancer will become very easy. Okay, so the answer for this question is, we are eliminating option number 3. It is option number A, because I told you that overactivity of TDP1 makes cancer cell resistant, not more sensitive, because it will repair the damage that has meant to kill them. Okay, coming to question number 16. TNBP, which is a gene editing tool that has been recently developed by ICAR. With reference to the TNBP recently developed by ICAR for genome editing in plants, consider the following statements. TNBP is a genome editing enzyme discovered in a highly radiation resistant bacterium. This is larger and more error prone than CRISPR-Cas9, making it less suitable for use in plant cells. TNBP can target specific genome resistance that traditional genomes like CRISPR-Cas9 and CRISPR-Cas12 cannot. It has shown successful application in important crops such as rice and Arabidopsis. See, this is a very tiny tool, and this is more efficient than CRISPR-Cas9 and Cas12 technology. So option 2 becomes wrong. So if you are going to eliminate option 2, you can easily arrive at the answer. That is option number A, 1, 3 and 4. Why? Because unlike CRISPR-Cas9, TNBP is much smaller and it is more precise, making it easier to use in plant genome editing. It can easily target the regions where this Cas9 and Cas12 cannot go. Okay, that is the main advantage of this particular TNBP gene editing tool that has been developed by ICAR. Okay. Question number 17. With reference to organ on chip technology, consider the following statements. Organ on chip devices are engineered using living human cells to replicate the functions of real organs. Yes. OOC is allowed for personalized drug testing using cells directly taken from the patients. Yes. OOC technology is primarily designed to replace prosthetic organs in clinical treatments. So this statement is wrong. OOCs help reduce reliance on animal testing and biomedical research. Yes. All the statements except statement number 3 is wrong, because the OOC has been developed to check the viability of the medicine that is being given to a patient targeting an organ. So this will actually mimic the real organ taken from the live cells of the patient. So this can be highly useful in personalized drug testing, but it is not designed to replace the prosthetic organs in clinical treatments. So eliminating option number 3, the answer is option number A, 1, 2 and 4, 1, 3. Okay. Question number 18. With reference to SNP chips developed for Indian livestock breeding, consider the following statements. These SNP chips, I have in detail discussed about the background of this SNP, and SNP chips are used to detect single protein variations in DNA that affects traits such as milk yield and disease resistant.
The GAW chip and Mahesh chip are variants developed for genomic profiling for Indian cattle and buffaloes respectively. True. As the SNP chip project is led by ICAR with support from Ministry of Agriculture. This is a question mark because it talks about the livestock. So the Department of Animal Husbandry has to be in place instead of this ICAR. SNP technology allows early identification of high potential animals, aiding in more efficient breeding selection. Yes, statement number 4 is also correct. So eliminating option number 3, we are having option A and option C. So you will have a dilemma in option number 2, the GAW chip and the Mahesh chip. Yes, GAW chip is for the genetic profiling of Indian cattle, whereas Mahesh chip is for the genetic profile of Indian buffalo. So the answer is option A, 1, 2 and 4 only. Yes, okay. The project is led by Department of Animal Husbandry and Dairying and it is not by ICAR. So see, when there is a question related to biotechnology, these are the small tweaks that we can expect in the actual examination because they will not twist much. Okay, so for this you will have to have a strong hold on the basics to eliminate the options. Even if you are not knowing the right answers, this will help in eliminating the wrong options so you can easily or comfortably arrive at the right option. Okay. Question number 19. With reference to genomic selection techniques in crop improvement, consider the following statements. Genomic selections involve using DNA markers across the entire genome, just note down the word, across the entire genome to predict the breeding value of a plant. Unlike traditional marker-assisted system, genomic selection relies on known gene trait associations for accuracy. Genomic selections can accelerate crop breeding cycles and improve traits like drought tolerance, yield and disease resistance. It eliminates the need for phenotypic data in the entire breeding process. So you have to focus on the extreme statements that is being mentioned on statement number 1 and statement number 2. See, it is not always that the extreme statements are wrong. Sometimes the extreme statements may also be right, but that is purely based on the context that we are discussing. Okay, so the answer for this question is option number A, 1 and 3 only. So here this extreme statements becomes wrong. Why? Because unlike the here the first statements becomes correct. As I told you earlier, the extreme statements always are not wrong because yes, genome selection involves using DNA markers across the entire genome. Okay. Second statement, why it is wrong? Because unlike market assisted selection, this genomic selection does not rely solely on the known gene traits because the question has been given reverse in the statement. Okay, unlike traditional marker assisted system, gene selection relies on known trait. No, it uses data from across the genome variety. Okay, why statement number 4 is wrong? We know that it is not from the entire process. Okay, while genomic data is powerful, phenotypic data is still essential for model training and validation in genomic selection. So question number in this statement number 4 becomes 4 because it does not eliminate the need for phenotypic data in entire breeding process, because see in question number 3 statement number 3 it has clearly mentioned that phenotypic data is necessary. So statement 4 becomes wrong. So 1 is correct, 2 is wrong, statement number 3 is correct. So answer is option number A, 1 and 3 only. Coming to the last question of this particular session.
With reference to the use of Wolbachia bacteria in mosquito control, consider the following statements. Wolbachia is a naturally occurring bacterium introduced into Aedes aegypti mosquito. We all know that the target organism for this particular bacteria is Aedes aegypti mosquito to help curb the spread of diseases such as dengue and Zika. The primary mechanism led by Wolbachia reduces mosquito population by altering their feeding behavior and reducing reproduction rates. No. Wolbachia infected males cause cytoplasmic incompatibility. This is the important point, by which when mating with unaffected females leading to non-viable eggs. See, this Wolbachia bacteria will enter into the egg of the organism, where it will reduce the possibility of hatching eggs. That is called cytoplasmic incompatibility. Wolbachia can block the replication of certain virus within the mosquito, limiting its disease transmission to humans. I hope you would remember a image where I was discussing about how this transmission of a virus is prevented from one person to another. Okay, so the answer for this question is option number A. Statement number 2 is wrong. Why? Because it does not alter the feeding behavior. Its main effects are cytoplasmic incompatibility. That means it will make the cells not to reproduce. Okay, sorry, eggs not to reproduce. Thus, by reducing the population gradually, and eventually it will lead to the decline of the mosquito population. Viral inhibition not reduce reproduction by behavior change. Okay, so this is about this Wolbachia bacteria. So guys, these are the important topics, and these are all the MCQs based on those important topics. So kindly revise the video where I have discussed related to the concepts behind the topics, and from which we have made this MCQ. And I also request you all to watch the video on the genetically edited rice varieties which have been posted in the Clarity for Sure YouTube channel, where I was discussing about the process of this genetically edited rice, and also I discussed about the difference between the gene editing and genetically modified organisms. So coming to the end of this particular session, I hope the session was useful to you and it was informative, and we have also learned some of the elimination techniques and how to eliminate the statements based on the basic knowledge we have. So revise this lecture once, revise all the 3 lectures related to the biotechnology one, so that you will be able to answer the questions in the upcoming examination with utmost ease. So thank you so much for watching this video. I will meet you with yet another session. Until then, bye bye, take care, all the very best for your upcoming examination.